Good morning. It's a pleasure to be here talking to you today about our work on simulating proteins and particularly enzymes, as I'll talk about. As you'll see, the title's a bit different. I've slightly altered it. I'm not going to talk so much today about receptor activation, but rather about the use of interactive virtual reality for problems of structure-based drug design. I thought that's particularly interesting and potentially relevant for the intersection with AI. So I'll start off by talking about some simulations of COVID targets. So obviously this is very much on everyone's mind in the times of the pandemic, and simulations, particularly molecular dynamics simulations, have really come to the fore during the pandemic, demonstrating their worth in characterizing targets, SARS-CoV-2 proteins and other facets of the virus, and how their structure and dynamics relates to their biological activity. So specifically for COVID, simulations, molecular simulations, are contributing in a number of different ways. One is through identifying potential drugs. Those may be existing drugs that could be repurposed, or the design and development of novel inhibitors of SARS-CoV-2 proteins. Simulations are also helping with the design and development of vaccines, for example, helping to identify the stability and test the stability of vaccine candidates, looking at the structure of the virus and the interaction of viral proteins with, for example, human receptors, and in understanding the effects of mutations, which is obviously a very current problem with the emergence of various variants across the world. So an example of how simulations are helping with understanding the SARS-CoV-2 virus are simulations we've done of the SARS-CoV-2 spike protein. So working with Imre Berger, Christian Schafitzel and others in Bristol, what we were able to do is to identify a novel binding site within the SARS-CoV-2 spike protein. This is the protein that interacts with human receptors as the initial part of infection. And what Imre and Christian and others were able to do was to identify a pocket here within the spike that binds linoleic acid. And that linoleic acid, which is familiar as a component of sunflower oil, stabilizes the spike in a locked, closed and non-infective form. So it prevents the receptor binding domain from opening and engaging with the ACE2 receptor. So what Debbie Schumark and others were able to do, and Charlie Colenso with Jim Spencer, was to use MD simulations and docking to identify ligands that bind at this site, at the site where fatty acids bind, such as linoleic acid, with the interest really of preventing this opening. This is the receptor binding domain. When it opens in the spike, it's able to engage with the ACE2 receptor, and this begins the process of infection, triggering conformational changes in the spike protein itself before entry into human cells. So here simulations were helpful, first of all, in identifying linoleate, a deprotonated form of linoleic acid, as the ligand that binds to this fatty acid pocket within the spike protein, and helping to show in complement to experiment that it stabilizes this closed, locked form of the spike, which is less infectious. This is an interesting pocket because potentially it's a drug target, obviously. And what simulations, so docking and then MD simulations, showed is that other ligands may bind at this site. This is a hydrophobic and potentially druggable site. And what we found is that various vitamins and potentially some retinoids and steroids may bind at this site. So vitamin D, vitamin K appear to have some affinity within our computational models, within our molecular simulations. And potentially this may be a way in which those vitamins and perhaps other compounds could affect and perhaps reduce the infectivity of the virus. Another example of how simulations are helping, and identifying some interactions which may be of biological relevance and novel interactions is work we've done looking at the potential interaction of the spike protein, again, of the SARS-CoV-2 virus, in this case with nicotinic acetylcholine receptors. And what the simulations indicate is that a particular region of the spike on the side here, this is the whole spike protein, and this is the head region, which I've shown you before. And what the simulations indicate is that this region of the spike may have affinity for the binding pocket of nicotinic acetylcholine receptors. So we've done MD simulations which suggest that this spike peptide is not only solvent accessible, but it has the capacity and the affinity to bind to the binding site of nicotinic acetylcholine receptors. And if this is indeed the case, then this may play a role in disease pathology. And I think this is something that's worthy of further investigation. And here's a model we've created of the spike protein interacting with the nicotinic acetylcholine receptor. This is work of Sofia Oliveira. And what this shows is that the interaction that the simulations predict is feasible with the spike protein in a non-parallel, effectively orthogonal arrangement to the nicotinic acetylcholine receptor. Another important and promising target for COVID is the main protease of the SARS-CoV-2 virus, this heart-shaped object shown here. This is the crystal structure of the main protease, and this plays an essential role in the viral life cycle in processing viral polypeptides so that they can be cleaved and form their functional forms. So blocking this enzyme, inhibiting its action, would be a very good way to inhibit and block the viral life cycle and perhaps to develop antiviral drugs. And for the main protease, a very promising route to new antivirals is covalent inhibitors. So inhibitors that covalently bind to their main protease target that form a chemical bond to it. And clearly there's a need to understand the chemical reaction and the features and factors that determine the reactivity of inhibitors within the protein. And to do this, we've used QMMM methods. So this is work led by Vicent Moliner and Cassius Fidorek in Spain in collaboration with Alia Lodola. And what they've done is to model not only the reaction of natural substrates in the enzyme, and I'll come back to those later in the talk, but also to look at covalent inhibitors and their mechanisms and how they react in the protein and what determines their reactivity, not only in the forwards direction in forming the bound complex, but also in the reverse direction. So in these QMMM simulations, what Vicent and Cassia were able to do was to study the reactivity of a known inhibitor, a known covalent inhibitor, this N3 compound shown here, and then to look at, by simulation, the reactivity of potential variants, synthetically accessible variants, which could have different reactivity. And what they were able to show is that these different variants have quite different reactivity in terms of the reversibility of their reactions. So these postulated or proposed compounds B1 and B2 are quite different in how reversible they are, how quickly they would come off the enzyme, how quickly the covalent complex would be broken down. And that reversibility is very important because that would modulate the pharmacokinetics of the drug, if it is a drug, the pharmacokinetics of the inhibitor, and that may have important consequences and advantages in terms of its action, for example, in the body. So molecular simulations have really shown their value in understanding SARS-CoV-2 targets, and very early on in the pandemic there was a recognition across the community that there was a need to share these data, to share our models, to share what was coming out of simulations, to deal with these complex targets and resolve some of the challenges from simulation, and to connect potentially these data with different sorts of data. So how do you connect simulation data to biological or even clinical data? There's a need to share simulations. These are complex simulations with many variables and many uncertainties in many cases because of the complexity of the protein targets. So there's a need really to test the models. And then when we have these simulation data, let's make them available to the experimental community, to the biomedical community. And as I say, very early on, several hundred groups across the world signed up to a pledge to share data for SARS-CoV-2 simulations, for the simulation of these proteins. And I think this is not only valuable for the pandemic, but this is something that we should be doing as a community in future. Really, there's a need to share not only the outputs of simulations, but the input, and to share the data in great detail, to share in atomic detail the results of our simulations. And this is, of course, something where there's potential for learning, excuse me, for the use of AI, machine learning, to extract more value from not just one simulation, but many. So in the title of my talk, I've talked about multiscale methods. And this is really a general phenomenon across biology and modelling in biology, in that the problems we're interested in generally involve many different length scales and time scales. That's quite obvious. And it's also equally obvious that many drugs are small molecules binding to large molecules. And of course, the binding of drugs has macroscopic effects. Similarly, chemical changes to biological macromolecules have effects on the scale of much larger dimensions of the scale of organelles, cells, organs, and even organisms. So a general problem and need in simulation for biology is for multiscale modelling, methods that are capable of combining different levels of description together in perhaps a single simulation or in coupled simulations. Perhaps the most established type of multiscale modelling in molecular simulation for biomolecular problems are combined quantum mechanics and molecular mechanics methods. These became respectable really in 2013 with the award of the Nobel Prize to these guys down here, Karplus, Leverett and Varshall. And the essence of QMMM modelling is very simple, in that a small region around the active site where the reaction takes place, for example in an enzyme, is treated by a quantum mechanical method, an electronic structure method, and this is coupled to an atomically detailed but simpler description of the protein and solvent environment treated by a molecular mechanics model. QMMM methods are now quite well established in computational biochemistry, and particularly for examining enzyme catalytic mechanisms. Here are two examples, charismatic mutase and para-hydroxybenzoate hydroxylase, both quite long-winded names. These are well-established mechanisms, a glycine rearrangement and a flavin-dependent hydroxylation and aromatic hydroxylation. And here we can be confident of the chemical mechanisms and use QMMM modelling to really examine features of catalysis, how the barrier is lowered in the enzyme, and also to test aspects of the QMMM simulation. So one of the reasons for doing a QMMM simulation of a reaction in an enzyme is to understand the reactive species, of course, to understand those species which can't be easily studied by experiment, such as the transition state for a reaction. And this shows the transition state from QMMM modelling for one of those enzymes, for PHBH, para-hydroxybenzoate hydroxylase. And here you see an OH group in flight from the hydroperoxyflavin cofactor, forming a bond with this aromatic ring of the para-hydroxybenzoate substrate. And this is a transition state, so a partially broken and partially formed bond. And what's interesting here is that in the transition state, we see that it is stabilized by a hydrogen bond to the backbone carbonyl of a proline residue, and this interaction is only formed in the transition state. So this is an example of an interaction which is catalytic. It lowers the barrier to reaction by stabilizing the transition state specifically, as revealed by these QMMM calculations. So as I mentioned, we can use these two enzymes, charismatic mutase and para-hydroxybenzoate hydroxylase, to test different flavors of QMMM calculations. And this is a table beloved of computational chemists, lots of acronyms. But what this shows in essence is that the barrier to reaction is not surprisingly dependent on the level of QM theory used in the QMMM calculation. And at the highest levels of theory, coupled cluster, which are as we know from first principles, the best of these QM methods, we can calculate barriers to reaction which are, for these two enzymes, in agreement with experiment to within chemical accuracy, so-called chemical accuracy, to, in other words, 1 kilocalorie per mole. And that shows that we can now predict barriers for reactions in enzymes with about the same level of accuracy as an experiment can achieve. And that means that we, at the highest levels of QM theory, we have calculations of reaction mechanisms in enzymes that are truly predictive in terms of mechanism. So the calculations I've shown there use local approximations, which are a little bit fiddly to apply. A more straightforward way to reach high levels of accuracy in QMMM calculations is through an embedding approach. This is a projector-based embedding approach developed by Fred Manby and Tom Miller and others. And what we've done is to apply this in a QMMM context, where we treat a small region within the reacting region by a high-level ab initio method, such as coupled cluster theory, within a larger QM region, treated, for example, by density functional theory, embedded within an MM environment, an MM description of the rest of the protein and the solvent environment. And why is this important? Well, density functional theory calculations are very variable in terms of the results that they can give, and very sensitive in many cases to the particular choice of the functional that's used in the calculation. So this shows a reaction barrier for the reaction in charismatic mutase, and this is the barrier calculated with various different perfectly respectable and good density functionals. And you can see that the barrier is very sensitive to the choice of the density functional in the QMMM calculation. So these calculations are not really predictive. On the other hand, when we embed within these calculations a small region treated at a high level of QM theory, in this case MP2 calculations, SCS MP2 calculations in terms of the details of the calculation, what you can see is that the sensitivity to the functional disappears. So using an ab initio method embedding in a DFT region gives us a method which is potentially predictive of barriers and therefore of mechanisms in enzymes. So an area where we've used QMMM calculations to look at mechanisms and make predictions about activity is antibiotic resistance. And this is a problem which certainly has not gone away during the pandemic. In fact, it's got worse, for example, because of the prescription of antibiotics to COVID patients. And this is a problem, antibiotic resistance is a problem that should worry all of us because it threatens many aspects of modern medicine. One particular cause of concern is the resistance to carbapenems, so-called last resort antibiotics, among particularly gram-negative bacteria. And these bacteria, which can be found in the gut, if you end up with an infection from one of these carbapenem-resistant enterobacteria, then there may be no effective treatment because of the resistance of the bacteria to carbapenems and to other antibiotics. And in that case, your odds of survival are significantly reduced. And this is a major cost and a major threat to healthcare systems across the world. So a major cause of antibiotic resistance, resistance to carbapenems and other beta-lactam antibiotics, is beta-lactamase enzymes, enzymes expressed by bacteria that break down beta-lactam antibiotics. And we've looked at this, looked at the reactions of beta-lactam antibiotics in beta-lactamase enzymes using QMMM simulations. And using QMMM simulations, we think we've identified the mechanism of reaction, which involves acylation and deacylation. And a QMMM calculation, as I've mentioned, you can calculate, for example, the transition state along a path, and you can calculate the free energy of reaction using molecular dynamic simulations with a QMMM potential energy function. So the particular problem of resistance to carbapenem antibiotics is relatively recent. So there's been resistance to other beta-lactam antibiotics, such as penicillin, for many years, clinically relevant resistance. And that's been due to some of these enzymes shown up here, for example, TEM1, which have been the cause of significant resistance to beta-lactams for a long time. More recently, enzymes have been widely occurring in bacteria that are able to break down carbapenem antibiotics efficiently. And they're able to do so by deacylating rapidly. So these enzymes up here are inhibited by carbapenems quite effectively. There's a covalent complex formed, and it only breaks down quite slowly. Whereas these enzymes down here deacylate rapidly. The covalent complex breaks down very rapidly. So using our understanding of the mechanism of reaction from previous QMMM simulations, what we've done here is to use QMMM molecular dynamic simulations to calculate the free energy barrier for this process of breakdown of the acyl enzyme, of the covalently inhibited enzyme. And what these simulations show is that we can distinguish between the enzymes that are inhibited and those that are not inhibited. So the QMMM calculations are capturing the essential details, the essential physical basis of this activity, of this antibiotic resistance activity. So we can distinguish between the older enzymes with slow deacylation that have high barriers to reaction from those newer enzymes which are resistant, that break down the acyl enzyme rapidly. And we can do calculations now by either calculating a full free energy surface or just a single one-dimensional free energy profile. We can significantly reduce the time taken to calculate these barriers in the MD simulations. And by even reducing the amount of sampling, we can go to even smaller lengths of simulation. So this is calculating the energy profile, but doing the opposite of what you'd normally do. So reducing the amount of sampling at each point along the reaction coordinate. And we still have predictive simulations here. So the simulations are capturing the difference and the causes of the differences in activity between the resistant and non-resistant enzymes. What causes this? And I think this is a challenge for machine learning, for AI. How can we identify the features and factors that are responsible for this activity? The causes are there within the simulations, within the structures produced by these QMMM-MD simulations. They are quite complex, multifactorial, and so we need sophisticated approaches to extract the fundamental determinants of antibiotic resistance. And similarly, looking at another class of beta-lactamase enzyme and a slightly different sort of antibiotic resistance, so specifically the activity of class D beta-lactamases as opposed to the class A enzymes I've just been talking about, and looking at their activity against cephalosporins. And here we were able to compare enzymes that are quite active against these species against enzymes that are not so active. And again, the QMMM calculations reproduce the difference in barrier between the two different sorts of enzymes. So they're capturing the difference. And here the activity of the enzyme against the antibiotic seems to depend on very subtle changes, small changes in the active site, small changes in hydration specifically. So how many water molecules are in the active site and how they stabilize the reacting groups. And those are features which potentially could be learnt from the simulations that we've performed. So here I'm going to switch gears slightly and talk a little about receptor activation and how simulations can help to reveal mechanisms of receptor activation. So we've looked at nicotinic acetylcholine receptors. I mentioned those previously as potentially interacting with the spike protein of the SARS-CoV-2 virus. And what we've done here is to use nonequilibrium MD simulations to identify a general mechanism of signal propagation. What we're aiming to do here is understand how the binding of a small molecule, in this case nicotine, is transmitted through this rather complex nicotinic acetylcholine receptor to activate it, to allow the passage of ions and the transmission of a signal. Simulations of these receptors, two different types of receptors, the alpha-4 beta-2 and alpha-7 receptors, which are complex proteins embedded in a membrane, I've identified what we think is a general mechanism for signal transduction in these receptors, or at least the initial stages, of how a ligand, small molecule ligand such as nicotine, binds to the binding site of the receptor, and how that binding triggers conformational changes which lead eventually to the opening of the receptor and the passage of ions across the membrane. Simulations reveal is that the binding of the ligand causes conformational change which is transmitted through specific networks in the protein, and it reveals the communication within the protein. So how the protein architecture allows for the transmission of a signal from a binding site to distant regions. And we think this particular structural mechanism shown here is general not only perhaps for nicotinic acetylcholine receptors, and the specific structural motifs are shown here, but also potentially has relevance for other receptors that fall into this cis loop family. So eventually the signal from the binding site is transmitted, we think, to the cis loop, so-called, and the M2, M3 loop, leading eventually to the opening of the receptor and the transmission of a signal. So as a slight aside, here's an artistic representation of that mechanism from machine learning. So a model's been trained here to learn from Georgia O'Keeffe paintings, and given a series of structures of nicotinic receptors, Dasha and Rob have produced this rather beautiful movie, which is a representation of those structural changes I've just shown you from non-equilibrium MD simulations. So if you like, it's from machine learning, a picture of how a nicotinic acetylcholine receptor works in the style of Georgia O'Keeffe. So what are the details of these non-equilibrium simulations? Well, obviously there are many different flavors of non-equilibrium simulation. What we've done here is apply an approach which was developed by Giovanni Chicotti and others, and what it involves is taking an equilibrium trajectory, and from that equilibrium molecular dynamics trajectory, sampling configurations at regular intervals and perturbing each of those similarly. And the perturbation in the case of the nicotinic acetylcholine receptors was the removal of a ligand such as nicotine. And then what we do is monitor the changes in the system in response to the perturbation, look at the difference between the equilibrium and non-equilibrium simulations, and by averaging over many pairs of simulations, you remove the noise and get a statistically significant measure of the response of the system to the perturbation. So we've used this non-equilibrium molecular dynamics approach to look at beta-lactamase enzymes specifically class A beta-lactamase enzymes of the type I mentioned earlier, and to look at the effects of allosteric ligands. So to look at how a ligand binding a long way from the active site can affect the chemistry of reaction at the active site. So how does binding of a potential inhibitor far from the active site modulate the activity of the enzyme? And these simulations have really revealed that indeed there is communication between the allosteric site and the active site in these enzymes. So this shows one of these antibiotic resistance enzymes. This is specifically KPC2. And what this movie will show is how changes at an allosteric site, distant from the active site, are transmitted to the active site. So here we're doing non-equilibrium simulations, starting from an equilibrium MD trajectory, where we remove this allosteric ligand and see what the changes are in response within the protein. Keep your eye on this omega loop, which is important in activity, as well as the active site over here. So as the simulation proceeds, and this is averaging over many non-equilibrium simulations, you see the effects of the perturbation at the allosteric site transmitted through loops of the protein, particularly in this case, to functionally important regions of the protein, particularly the omega loop. So this shows that there is communication between distant binding sites and the active site along defined pathways within these class A beta-lactamase enzymes. So these non-equilibrium MD simulations identify pathways within the protein by which effects are transmitted from distant sites, allosteric sites, to the active site of the enzyme. And what was particularly interesting is that the pathways that we've identified involve positions that are clinically relevant. In other words, mutations on these pathways affect the enzyme's activity. They are related to the spectrum of activity. So different enzymes of these classes have different activity against different sorts of antibiotic. And it appears that this activity relates to their allosteric behavior. So these simulations are identifying positions of clinically relevant variants, and these positions of mutation apparently alter the allosteric behavior of the enzyme, and by doing so they alter its spectrum of activity against different antibiotics. So this idea that distant regions of an enzyme may affect its catalytic activity relates to that fundamental question of why are enzymes big? So enzymes are large molecules, and a really striking correlation is the fact that for more difficult chemistry, nature has evolved larger enzymes. So there's a reasonable correlation between the acceleration of the reaction, so how difficult is a chemical reaction, how high is the uncatalyzed barrier relative to the catalyzed barrier, with the molecular weight of the catalytic domain of the enzyme. And why is this? Well, in part it's due to the fact that you need folding energy to organize an active site, and that is a pre-organization effect that gives rise to catalytic proficiency. But it also means that there's potentially a heat capacity effect, and we think this is important. We think that evolution has acted to modulate the activation heat capacity of enzymes to tune their temperature dependence. It also leads in some interesting directions. For example, it suggests the heat capacity effect suggests that proteins might offer evolutionary advantages over, for example, ribosomes. So why did we move from an RNA world? Well, potentially this may be in part due to the fact that the larger heat capacities of proteins allows for more change in heat capacity, and that may improve their evolvability. They may be more evolvable than RNA molecules for this reason, though that's speculative. But the heat capacity effect, we think, is fundamentally important in the adaptation of enzyme catalysis. So many enzymes show rate profiles that look like this. They're curved, so there's a temperature of maximum activity, and above that temperature, activity drops off. The textbook explanation of this is that the enzyme unfolds and so activity is lost. But for many enzymes, perhaps particularly most obviously those from cold-adapted organisms, activity is lost far earlier than unfolding takes place. This curvature of activity, of the temperature dependence of activity, can be explained by a negative activation heat capacity, which we have described within what we call macromolecular rate theory, which is really just transition state theory, but taking into account the fact that the heat capacity may change between the ground state and the reactant state. And this curvature, this MMRT behavior, is apparent on the large scale for processes such as plant respiration measured at the field level, for example. So this is potentially of wide-ranging significance. We can use molecular dynamic simulations to calculate the activation heat capacity of enzymes by measuring the fluctuation in the energy in the reactant state and the transition state or a transition state analog. And what we find is that simulations give activation heat capacities that are in good agreement with experiment, showing that there is indeed a tightening up of the enzyme in the transition state ensemble. And these simulations show that parts of the protein distant from the active site play a significant role in modulating the activation heat capacity. So the fluctuations of distant parts of the protein contribute to the overall response. So the whole architecture of the protein is responsible for the change in heat capacity, and it's something on which evolution can act. And we've looked at these effects in the context of enzyme design, so looking at the Kemp elimination reaction, a reaction that's not naturally catalyzed specifically by nature, so there are no natural enzymes optimized for this reaction, but a number of unnatural catalysts, artificial protein catalysts, have been designed and developed for this reaction. Generally, they have to be optimized by directed evolution to produce a good catalyst. And what's really striking is that when you evolve catalytic efficiency, going from a designed protein to an evolved protein catalyst, you see the emergence of curvature in the temperature rate profile. And this is most unexpected, but it shows that somehow evolution is driving the appearance of macromolecular rate theory type behavior, of the appearance of a negative activation heat capacity. And just as with the natural enzymes, we can do simulations of the transition state ensemble and the ground state ensemble in the designed and evolved catalysts, and from those simulations calculate the activation heat capacity. And what the simulations show is there is indeed a tightening up of the transition state ensemble, specifically only in the evolved catalyst. And what these simulations show by analysing the correlations in the different species is that the evolved protein has networks, correlated networks in its transition state ensemble, so specifically in the reactive form of the enzyme, which don't exist in, or are much weaker in the design. So evolution has introduced correlated networks, and those networks appear to predict the positions of mutation. So those networks appear to be a guide to engineering enzyme activity. So I think here really the challenge is to use simulations to guide enzyme design and to learn what it is that evolution is introducing, and specifically to understand that it is by targeting the transition state ensemble and the difference in dynamics of the transition state ensemble compared to the ground state that we may capture some of those effects. So I'd like to finish off by talking quickly about what I think is a very interesting and transformative way of interacting with simulations, and that is in virtual reality. And I think this provides potentially a very fertile interface with machine learning and AI, that we can learn from how humans interact with simulations in VR. So we've used the Narupa interactive MD framework developed by my colleague Dave Glowacki in Bristol to look at structure-based drug design problems. So here is Becca Walters, a PhD student who works with me and Dave. And what she's doing here is using interactive molecular dynamics in virtual reality to bind the drug oseltamivir, Tamiflu, to its target influenza neuraminidase. And you'll notice that this is a flexible system. So Becca is interacting here not just with a static model, but with a molecular dynamic simulation. And you can see how the protein is responding to the presence or absence of the drug, because it's able to respond flexibly and change its conformation dynamics in response. And what Becca and Helen Deeks and others were able to show is that we can use interactive molecular dynamics in virtual reality to predict the binding modes, poses of drugs bound to their protein targets, that even non-specialist users can recreate the structures of drug complexes using interactive MD in VR. So this gives a user's view of interactive MD in VR. What Helen is doing here is removing a small molecule ligand, benzamidine, from its protein target, trypsin, in a molecular dynamics simulation. So again, you can see the protein and the drug wiggling around because this is a dynamic simulation. And so the protein and drug respond sensibly and flexibly to each other and to the process of binding and unbinding. And you can see here how Helen uses the menu to control aspects of the simulation and how she's interacting with it and change the view. And we've used these interactive molecular dynamics simulation techniques to look at some of the targets I talked about at the beginning for SARS-CoV-2, and specifically the main protease. So what Helen and Becca have done is to use interactive MD in VR to look at the binding of flexible molecules to the main protease. So the job of the main protease in the viral life cycle is to bind and chop up the polypeptide that then goes on to form the components of the virus and all its stage of its life cycle. And what Becca's doing here is studying the binding of peptide to the main protease. Again, you can see this is a fully flexible system. So the protein is moving. The long flexible protein chain that it chops up is also flexible, dynamic, and moving around. And Becca here is manipulating it and finding how it binds into the active site. So how does a long flexible molecule bind into a plastic protein system like this? And interactive MD in VR offers a number of advantages. The ability to manipulate molecules in a way that you would manipulate a macroscopic object, to walk around the problem so that you can see different aspects of it and experience and understand the problem in 3 dimensions. And when mounted in the cloud, Interactive MD in VR also offers possibilities for virtual collaboration. So here Helen and Becca are working together virtually on the problem I've just talked about, on the main protease of SARS-CoV-2. And here they're able to manipulate the same molecular system, the same simulation at the same time, and to see what each is doing, and for each of them to manipulate the binding and understand what's going on, and to collaborate and share that information by coexisting in the same virtual molecular space. The need for virtual collaboration really has been highlighted by the pandemic, and this sort of VR technology provides a new route to virtual collaboration for problems in structure-based drug design and indeed in other aspects of molecular science. Interactive virtual reality also offers obvious advantages and attractions for education. The ability particularly to work together and demonstrate molecular problems in VR, I think, is really quite exciting. And in Bristol, what we're doing is we're teaching undergraduates about enzyme catalysis using interactive MD in VR. So I mentioned charismatic mutase before, so we teach them how to carry out a Claisen rearrangement reaction using a QM method, a density functional theory method, approximate DFT method in VR, and also give them the opportunity to study docking, the binding of a substrate into an enzyme using interactive MD in VR. And perhaps not surprisingly, students found this much more engaging than traditional computational chemistry classes, and we believe that this leads to improved educational outcomes. And of course, we can learn from how the students are learning. We're able to capture the trajectories, and this could lead to, for example, crowdsourcing of problems in drug design. So I'd like to finish off by thanking the people who funded this research, both in terms of money and in providing computer time. Thank my group, shown here a couple of years ago in pre-pandemic times when we were all able to meet physically. To thank everybody who's assisted with technical aspects of the research in many different ways, from software engineering and the support of our Advanced Computing Research Centre in delivering this research and allowing us to carry out our simulations, and the provision of computer time on the Archer 2 and Archer systems from EPSRC, cloud time from Oracle, and thanking the University of Bristol Emergency COVID Research Group for support. And I'd like to thank all my collaborators in the research that I've talked about here.